Adrian Harwood. I'm the other co-organiser from the BSCB, and I've got my glasses, so I get this job. Okay, so the Hooke Medal is awarded by the BSCB each year to recognise an emerging leader in cell biology. Okay, the award is named after Robert Hooke, okay, the eminent 17th century natural philosopher. That's when scientists were natural philosophers, who is obviously known for his pioneering work in microscopy, and particularly the first book of microscopy images. And the medal design was designed by Brad Amos. I won't get it out. I'm sure Anne will show it you later if you ask her nicely. It basically shows his original microscope on the one side, and then cork cells on the other side, and that's where the word cell comes from, which we wouldn't have a society without him, I think. Okay. So the award is given to an individual who, although within their first 10 years of setting up their own lab, has already made an outstanding contribution to the UK cell biology field. Okay? And so it's a great pleasure this year to award the medal to Anne Bertolotti. So Anne obtained a PhD at Strasbourg University with Pierre Chamon. She and Laszlo Tora. She then pursued a postdoc in David Ron's lab at Skirball in New York, and then returned to France before being coaxed, I suppose, or persuaded to come back to the UK, to come to LMB in Cambridge, where she's been since 2006. Okay, so she's made important contributions to our understanding of the mechanisms that underlie the deposition of proteins, unfolded proteins in the cell, but, which obviously you know is sort of associated with a whole series of sort of protein aggregate diseases, such as Alzheimer's and Parkinson's. And more importantly, she's interested in the mechanisms of how to survive this process. She was elected an EMBO Young Investigator in 2005 and an EMBO member in 2013. Okay, so it's a great pleasure then to award this medal to Anne, if you come here. And we've got to make sure we stand for the photographs. So do you want to go this side? And smile. Okay, so it's a great pleasure. Okay, and just to prove there's nothing free in life, she's now got to give you a seminar for half an hour. Thank you.
Thank you very much. Well, it's obviously a great honor to be here today, about to give this Hooke Medal lecture, and I was absolutely thrilled to be chosen for this and to join a rather long list now of really great scientists, so thank you very much. And as a token of my gratitude, I brought you today the original drawings of the Hooke Medal by Brad Amos, who, as you may know, when he's not busy making drawings for medals, also made some slight improvements of the original microscope that Hooke designed. I think an award brings recognition, and that recognition goes to a field or a group of individuals rather than to one single person. And I would really like to take this opportunity to thank the people who have either inspired or contributed to the work I'm about to present. So I'd like to thank my mentors, my colleagues, and most importantly, all my group members. I've had, and I'm still very fortunate to have really, really great scientists with me. I have a group of really talented and enthusiastic people who have joined me and taken risks in basically exploring the unexplored and trying to push the boundaries of knowledge. So many thanks to them, past and present members of the lab. In my lab, we are interested in proteins that don't always behave well and have the tendency to misfold and aggregate. And these proteins really represent an important problem for cells and organisms, because the position of misfolded proteins is actually at the origin of a really broad range of human diseases, including the devastating neurodegenerative diseases which affect an increasing number of individuals in our aging societies. Now, as you may know, these diseases, Alzheimer's, Parkinson's, amyotrophic lateral sclerosis, prion disease, or Huntington's disease, are clinically different, but they share some common properties. one important property that I will remind you throughout this talk is that these diseases are late onset. They are diseases of old age. And the second shared property is that these diseases share a common histopathological hallmark, which consists in the deposition of disease-specific proteins into insoluble aggregates. Now, the disease-causing proteins, the major components of the disease characteristic deposits, have been identified in most cases over 17 years ago. And the disease-causing proteins are remarkably different. They don't share any sequence similarities. They are also very different in their native fold. However, they do share some common features. The disease-causing proteins are actually soluble proteins that are expressed throughout the life of individuals, and they are benign for many, many years. But at some point, late in life, they misfold, and this has a series of really catastrophic consequences for cells and organisms. And why this happens and how this happens is unclear. So really here we have identified what I think is an important problem, trying to figure out what are the mechanisms at the origin of these devastating diseases. And that's a good start. If we want to make scientific progress, the first thing we need to do is to identify a problem. So we got there, so far so good. But what's even more important is to try and find a way to solve this important problem. And in my view, this complex organismal problem is in fact at the origin a cell biological problem. And if we realize this, then when we can break this big problem into two questions, and that we can then hopefully solve. And so in my lab, we've been interested in trying to understand the mechanisms that govern the deposition of disease-causing proteins, because this is one of the earliest events in the disease process. And in addition, a related question my lab has been putting a lot of effort into is to try to identify strategies to help cells reducing the burden of misfolded proteins, with the view that if we manage to do this in cells, well, perhaps we can do that in mice, and if we do that in mice, well, perhaps we can do this in humans. So essentially, the work we do in the lab focuses on what I think are fundamental cell biological problems, with the view that our progress may ultimately benefit human health. And today, in today's lecture, I'll share with you our progress towards answering these two important questions. In the recent years, we've been really interested in amyotrophic lateral sclerosis, or Lou Gehrig's disease. And the reason for that is that we found that actually the characteristic clinical features of this disease were really inspiring and helped us understanding the mechanisms, the molecular mechanisms at the origin of this devastating disease. So amyotrophic lateral sclerosis is a fatal motor neuron disease, which is characterized by the degeneration and loss of motor neurons, and like many other neurodegenerative diseases, this starts in adult life, usually after 50 years of age, and it's really a dreadful disease which results in death. It's inevitably fatal due to respiratory failure. Now, what's really striking about ALS is that it's a really rapidly progressive disease. ALS kills patients. Half of the patients die a year and a half after symptoms onset, and this is really similar to prion disease, which are also really fast and furious diseases, but in sharp contrast to the other neurodegenerative diseases, Alzheimer's, Parkinson's, or Huntington disease, for which the disease duration is much longer and lasts for 10 to 20 years. So there's something there that we need to understand. Another remarkable feature of the clinical manifestation of this disease is that the clinical manifestations are heterogeneous. And this has been puzzling for years for clinicians, but actually this is due to the fact that the region of disease onset is variable. So there's bulbar onset, which causes speech or swallowing impairment, whereas the onset can also affect muscles in the limbs, so in the arm or in the leg. So it's a very diverse set of symptoms, but regardless of the site of onset, ALS always begins focally in one discrete body region, and that's in 98% of the cases. And then the symptoms inevitably progress to neighboring areas where they appear with decreasing severity. So this has suggested